Continuing on with the cell membrane, we'll now take a look at the movement of substances through the cell membrane as we go into the processes to allow for transport through the membrane, either into or out of the cell. And so the membrane proteins themselves, we talked last time and we saw that there were many different types of proteins that are embedded within the membrane, some of which are transport proteins. Those transport proteins can be simply small channels, and so they can allow for substances to simply move through the membrane itself. And so we can get water-soluble substances to come right through there and move on in. We can have gated channels, and so they can have a little gate on them. Sometimes those are closed, sometimes those are open, depending upon how the substance itself is moving through, what's activating those membranes, things like that. Depending upon the particular substance as well, and the particle that is coming through, things like them being charged in terms of those proteins can make a difference as well. And so sometimes charge within the cell can allow for that structure to be closed, and then we change the charge to a positive charge, and then it's going to open it up, and we allow that substance to then move through the membrane. Other substances, rather than in a sense kind of just having an open hole or an open pore in a sense within them, whether it is gated or anything like that, we can have carrier proteins, where now our substance is going to have to fit that specific space there. And so this is typically going to allow us to get larger molecules into the inside of the cell, but it has to fit that particular shape. And so each side of our protein has a half circle in it, and those half circles then in a sense kind of close around our molecule there. And once that molecule comes into our carrier protein, that carrier protein is then going to change shape and allow for the substances to come inside. Always going from an area of higher concentration towards an area of lower concentration. And so it's always following that concentration gradient moving through the membrane itself. Other proteins we mentioned before have things like receptors on them, and so they can bind up a particular substance, similar to that of like a lock and key. And so in order for that particular receptor protein to become activated, we have to have the exact type of a molecule come into it. And so it's going to then change something that's happening inside the cell that's there. It may even take one of those channel proteins themselves, and perhaps it has a binding site here for a particular molecule, and it's also got a little gate on it, and we have to get the right substance to come into there in order to get that gate to swing open. And so that's going to be determining when and how often that opens up. Other things we talked about on the surface of the cell, we have things like marker proteins. And so we have an embedded type of a protein there. It's then got its little glycocalyx that's there. This is going to be our kind of name tag for the cell. And so this is going to make sure that that cell is recognized as being normal, as being part of self, and that allows us to determine other cells that may have a different type of a protein that's there, a different type of glycocalyx. When it's there, it has a different shape to it, and our body is going to recognize that as being potentially foreign. And so it's going to destroy those cells, that bacteria, viral infected cell, tumor cell, whatever it happens to be, that has that changed or altered glycocalyx on its surface. Some of those proteins as well act as enzymes, and so they're going to start a chemical reaction faster, use that catalyst to get the chemical reactions going much more quickly, and allow us to make changes to the chemicals that are needed for that particular cell. Other protein channels have part of our cytoskeleton embedded into it, and so this can allow for the cell to maintain its shape, to be protective for it in terms of support, allow for the cell to move, all kinds of different categories for that particular cytoskeleton. And then we can help to hold cells together. And so in places where we have tissue that needs to be held tightly together, we can have special connections between the cells that are there and allow us to kind of hold them together, create a nice barrier in between them by simply, in a sense, kind of welding those cells together. And so we have these little adhesion proteins that are there with them. Some of them add more support to the cells. It all just kind of depends upon the cell itself. And so we have those cytoskeletons there in between the cells. And then we also have adhesion proteins that help to kind of bind them up and hold them together. When it comes to the movement of molecules, one of the things that we rely upon is diffusion. And diffusion is going to allow us to generate a process where we're going to want to always try and get the solution into a equilibrium so that we have the same concentration on either side of a particular membrane that's there. And so it's going to go from an area where we have a higher concentration. So if we put this little dye cube inside a beaker of water, initially we have high concentration in that little area, and it's going to want to dissipate and move throughout the beaker here until we have an equal concentration throughout the entire beaker. And so it's going to move with its concentration gradient. It's going to move down its concentration gradient, always going from a higher concentration to a lower concentration within whatever fluid we happen to be in at that time. Different types of substances can dissolve inside the water itself and create this diffusion that is there. And they need to be things that obviously like water, so things that are going to be hydrophilic are going to allow to be moved and kind of taken across that concentration gradient. So things like that dye, we can dissolve it inside there. Other things like electrolytes. So we have certain substances, and so when we put sodium chloride in water, we're going to dissolve it in there, and it dissociates into our sodium and our chloride ions inside there, and it can dissolve inside. Hydrophobic things, so if we were to dump some oil inside there, that oil would end up just kind of sitting on the surface of the water, and we wouldn't see any kind of mixture. No matter how much we mixed it up, we wouldn't be able to get the two to eventually dissolve together. Water does work kind of on its own to a certain degree, and so although water by kind of definition is water soluble, so to speak, and so it is water itself, technically it should not be able to get through this plasma membrane. And so it shouldn't be able to get through that membrane itself, because remember, on the inside we have our phospholipids in that membrane, and we have a hydrophobic center where we have all of those tails, and the hydrophilic portions in the heads are on the outside. So water likes the outside, likes the inside of the cell, but the innermost aspect of that membrane, technically in a sense, we shouldn't be able to get water to move through that lipid bilayer that's there, but it does allow for some of it to come through. The majority of it is going to come through special proteins called aquaporins. And so these special channels are there to allow for water to come through the membrane. And again, it's going to move down a concentration gradient. When we have, in a sense, more water over here compared to over here, so less water, more water, the water is going to want to move to the other side and come through to the other side there. At the same time, we can think about it in terms of solutes. And when we have a solute concentration that is high over here and a solute concentration that is low over here, again, the water is going to want to move over to this side to be able to dilute out that solute. So if we have certain substances that are there in a greater quantity on the inside of the cell, it's going to draw water towards the inside. And this concept is where we come into tonicity. And so depending upon the particular solution itself, tonicity is going to affect the volume of the cell, and so how big the cell gets. So does it blow up, or does it stay the same? Does it shrink? Things like that. It's going to affect where the solution goes. And so how it affects the cell itself is going to dictate the tonicity that's there. And what it comes down to is we have a membrane. So we have essentially a barrier that's there. And we have a concentration of solutes on one side. We have a concentration of solutes on the other side. And when we compare the two to one another, it's going to dictate whether, in terms of water, if that water wants to move towards this side or whether the water wants to move towards that side. And so we have, again, a semi-permeable membrane that's there, and that's going to affect the flow. In cells or in solutions where we have an isotonic solution, this isotonic solution, essentially the concentration of the solutes on the outside of the cell, so we'll put O there, and the concentration of the solutes on the inside of the cell are equal to one another. And so we have the same concentration on either side, and so basically the movement into the cell and the movement out of the cell are equal to one another. So there's no net movement really of water for those. So now when we start to look at the concentrations comparing the inside and the outside of the cell when we have a hypotonic solution, now our concentration of the solutes outside of the solution, outside of the cell, compared to the concentration of the solutes inside of the cell, we're going to see that outside of the cell we have less concentration than we have inside the cell. So the water is going to want to move into the inside of the cell, and so our outside concentration is lower. So the solute concentration outside is less than that inside, and so we have our water starting to move into the inside of the cell. As it moves into the inside of the cell, that cell may eventually get so much water inside of it that it undergoes hemolysis. And so the red cell itself is going to eventually burst open and be destroyed in that particular solution. If we, in a sense, reverse it, and now we have our solute concentration outside of the cell compared to our solute concentration inside of the cell, now we have a hypertonic solution. So outside of the cell, it's high concentration. Inside, it's low concentration, comparing the two sides. So our water is going to move out of the inside of the cell to the outside because it's wanting to go towards the higher concentration. And so we have this net movement where now the cells themselves are going to start to shrivel up or undergo crenation on there. And so the two of them are going to now work in such a way that we have a greater concentration outside of the cell than we have inside, and that allows for that net movement to essentially shrink the cell up, and they start to get all these little kind of pointy parts on them. But this time they don't necessarily become completely destroyed like they do in a hypotonic solution. Larger substances are going to potentially require some ability to use different channels to get things through there. And so depending upon what we need to move through, we may need to use different types of channels within that. Other times we're going to be able to move things through without any kind of larger channels. And so we get passive transport. Passive transport does not require ATP. So no energy is going to be utilized in this process in order to move them along across that membrane. So we're always going to go from a higher concentration towards a lower concentration. So it's just simply going to follow its concentration gradient as it goes. Depending upon the particular substance is going to determine whether we need a protein involved or not. And so things that can potentially get through the membrane are substances that have that ability to simply slip through that phospholipid bilayer that's there. And so things like certain gases, oxygen, carbon dioxide, substances that can go through there. We already talked about potential for water to make its way through. Since we have a membrane here, which is polar, we need to have non-polar types of molecules make their way through on there. So they've got to be lipid soluble types of things to make their way through that membrane So simple diffusion through the lipid bilayer. They have to be small, they can't have a charge. So all of our electrolytes are not going to be able to just simply slip through that membrane. They're going to require that we have the addition of having a protein channel there in order to allow us to make their way through the membrane itself. There. Another picture, just simply different animation or picture there. So things like oxygen making their way through, carbon dioxide making their way through the cells, oxygen moving in, carbon dioxide, our waste moving out of the cells. If we need to have a protein channel, now we can still have a process where we have no ATP involved, so there's still no energy involved here. And we have a channel that is simply open. It's allowing sodium molecules to move from an area where we have high concentration to an area where we have lower concentration. And so once again, it's just simply following that concentration gradient to move substances into the inside of the cell here. So it's a channel that is forever opened. It allows things to move through that particular protein. If that particular substance is slightly different in terms of its makeup, that's there. And so we have something slightly larger like glucose here. We may have to use a carrier protein. So now we get back to that specifics. It has to fit into that particular receptor in order to allow it to move in through that particular protein. And so that allows us to generate, once again, a concentration gradient where we have a large amount over here. We have, in a sense, nothing over here. So we want that glucose to move in through the membrane itself. Some of those channels themselves may be gated, and so we may need to have a different process occur in order to get them to open up. And so here the gate is opened, here the gate is closed, and depending upon what is going on with that particular protein that's there, we may have a charge differential. And so we may have a situation where we have a positive or negative charge within that area. Here we get the negative charge becoming a positive charge, and it opens up the gate. This is going to, once again, allow us to kind of control what's going through there. Other times we're going to use energy in order to move against a concentration gradient. So here now we're going to move from a low concentration towards a high concentration. And so we're going to move in the opposite direction that it normally would want to go. And so the driving force is going to be actually in this direction for the concentrations that are there. But because we're going to use ATP, we can pump things against that process. It's kind of like pumping water uphill in a sense. And so if we get the water all the way to the top of a hill, it'll want to come down following its gravitational pull. But in order to get the water up to the hill itself, we've got to impart some energy into the system itself in order to get it up there. And so once again, since we're going to need some energy, we're going to also need some carrier proteins. So this is going to allow us to move those substances across that membrane in that opposite direction of the concentration gradient that's there. And this allows us to kind of similar to a gated protein or a carrier protein, it changes the shape of the protein as our substance enters into it. Other times we get large amounts or large substances into the cell by using endocytosis. We can also remove large substances out of the cells via exocytosis. So active transport can be something similar to that we'll see in muscles and neurons, where we utilize a calcium pump. And so this is our calcium pump here. It requires ATP to be present in order to move calcium against the calcium concentration gradient over to the other side of the cell. And so we have now a larger concentration over here compared to over here. And so we're moving against that gradient that has been set up. Sodium potassium pumps utilize the same process itself there for it. It's one of our more important ATP using pumps. It's going to allow us to move sodium and potassium in opposite directions through the same pump. And so sodium is going to move into the cell, potassium is going to move out of the cell at a ratio of 3 sodiums for every 2 potassium. So we're going to get 3 sodiums into the inside of the cell, and we're going to move 2 potassiums outside of the cell. And in that process, we generate the need for ATP. So our ATP will get broken down into ADP and inorganic phosphate, and then we can recycle it from there within the cell. But this is going to be an important one that we'll see for both muscles as well as neurons as we move along. Larger substances, we talked a little bit about this last time in terms of endocytosis and exocytosis. So endocytosis is going to bring our molecules into the inside of the cell. So we get them into the cell. This may be larger material that's there. This may be substances that need to get to the inside of the cell within a larger quantity that is there. Other times we're going to move substances out of the cell, and we're going to do exocytosis. Both of these processes involve these vesicles inside the cell requiring ATP in order to generate their movement. So we have to have them there in order to bring them into the cells, through the cells, and then potentially out of the cell. So our same substance that we may have taken into the inside of the cell on one side of the cell eventually goes through to exocytosis on the other side of the cell and gets lost through the cell. Or if this was potentially like a white blood cell, and that white blood cell had consumed the bacteria, it destroyed that bacteria, it brought it in as a phagocyte or as a phagosome, and then eventually, now that it's all destroyed, it's going to get rid of that waste via exocytosis. And so that's how, for the most part, we move things through the membranes of our cells.